Hello everyone, and welcome to today's webinar in the SERDP and ESTCP webinar series. My name is Rula Adib. I'm a Senior Principal at Geosyntac Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. I will be facilitating today's talk. The webinar will consist of a brief overview of SERDP and ESTCP by Dr. Andrea Leeson, followed by a list of upcoming webinars in the series. After Andrea's opening remarks, we will transition to the technical portion of the webinar. Today's event features two presentations summarizing research on monitoring and risk assessment of environmental risks posed by munitions constituents in aquatic systems. Dr. Todd Bridges and Dr. Guido Tufo from the U.S. Army Engineer Research and Development Center will discuss evidence regarding environmental risks posed by munitions constituents in aquatic systems. Dr. Bridges and Dr. Lotufo's presentation will be followed by a brief question and answer session. Next, Mr. Gunther Rosen from the Space and Naval Warfare Systems Center Pacific in San Diego will talk about the validation of passive sampling devices for monitoring MCs in underwater environments. We will conclude the webinar with an interactive Q&A session with all of today's three speakers. The broadcast today will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to submit questions well in advance of the Q&A session. On this slide, we have provided a few suggestions in case you experience difficulties with the broadcast audio. Typically, any delay can be fixed if you refresh your screen or call into the conference line. However, if you continue to have problems with either the audio or the visuals, please submit a comment using the chat box in the left corner of your screen. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for Environmental Restoration. Andrea has been with SERDP and ESTCP since 2001, and before that, she served as a research scientist at Battelle Memorial Institute, where she conducted research on in situ bioremediation. Andrea?
Thanks, Rula, and welcome everyone to today's webinar. And what I'd like to do now is just go over a little bit about the programs before I turn you over to our speakers today. SERDP is the Strategic Environmental Research and Development Program and was established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and the EPA. And our mission is to identify and address high-priority environmental science and technology opportunities that focus on DoD requirements. We fund under SERDP both fundamental research as well as advanced technology development that ultimately impacts real-world environmental management. ESTCP is the Environmental Security Technology Certification Program, and here we demonstrate innovative environmental and energy technologies. These investments capitalize on past investments under SERDP or other research programs and are designed to transition our technologies out of the lab and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. SERDP and ESTCP are complementary programs with much of SERDP research occurring at the lab and pilot scale with occasional field efforts, while under ESTCP, demonstrations are primarily at the pilot and field scale, although occasionally we do support lab efforts here also. We have 4 program areas in SERDP and 5 in ESTCP. The Energy and Water program is only in ESTCP, while the other 4, Environmental Restoration, Munitions Response, Resource Conservation and Resiliency, and Weapons System and Platform, are SERDP and ESTCP programs that are managed jointly by a designated program manager. Our webinar today is focusing on research and demonstrations that were conducted under the Environmental Restoration Program area. And under environmental restoration, we essentially have 5 main areas of research: contaminated groundwater, contaminants on testing and training ranges, contaminated sediments, wastewater treatment, and risk assessment. Our webinar series, though, will be highlighting research and demonstrations from each of the 5 program areas. And as you can see, we have upcoming webinars that are covering a broad range of topics, including 1,4-dioxane cleanup. We have solutions for installation participation in energy markets and tools for risk-based UXO performance or remediation. If you go to the link shown here, you'll get more information about all of our upcoming webinars, and you can also get registration for the webinars through the end of May. I am also extremely happy to announce that we are once again holding the SERDP and ESTCP Symposium. It will be November 28th through 30th here in Washington, D.C. We will have registration information live very soon on our SERDP and ESTCP website, and I hope to see you there. I hope you enjoy the webinar today, and at this point I'll turn it back to you, Rula.
Thanks, Andrea, and it is now my pleasure to introduce our first 2 speakers, Dr. Todd Bridges and Dr. Guy Lotufo. Dr. Bridges is a senior research scientist and director of the U.S. Army Engineer Research and Development Center for the group dealing with contaminated sediments. He is also the program manager for ERDC's Dredging Operations Environmental Research Program. Todd has served as the PI on several research projects related to sediment toxicology, contaminant bioavailability, and sediment risk and assessment and management. He has published more than 60 journal articles and book chapters, as well as numerous technical reports on these subjects. Todd received his bachelor's and master's in biology and zoology from California State University in Fresno, and his PhD in biological oceanography from North Carolina State University. Dr. Guy Lotufo is an aquatic biologist with ERDC in Vicksburg, Mississippi. For the past 18 years, he has conducted basic and applied research for the Army, primarily in support of the contaminated sediment dredging and environmental quality technology program. He has maintained active collaborations with leading national and international research organizations and has authored 78 peer-reviewed papers and book chapters. Dr. Lotufo earned a bachelor's degree in biology and a master's degree in zoology from the University of Sao Paulo in Brazil, and a PhD in zoology from Louisiana State University. And with that, I turn it over to you, Todd.
Okay, thank you, Rula, very much. And on the next slide, you see a brief agenda for our presentation. I'll be giving some introductory remarks, and followed by Dr. Lotufo, who will be discussing the nature of risks for underwater munitions and the work that we've been doing over the last several years in this connection. The next slide, titled Underwater MM, Worldwide Issue. I'd first like to say that ERDC and SPAWAR, the 2 DoD organizations presenting our work today, have been conducting research and producing science on the environmental fate and effects of explosives for more than 25 years. And for most of that time, the ERDC and SPAWAR have been collaborating on this topic, particularly in regards to underwater munitions. And the projects we're presenting today in many respects represent a culmination of these years of work and literally dozens of research projects, including many research projects that have been sponsored by SERDP and ESTCP on this topic. So as a result of military training and weapons testing activities, munitions are present at current and former U.S. Department of Defense sites in underwater environments. And I should add that there are many countries around the world who have similar sites and similar challenges related to their own activities. Many active and former military installations have ranges and training areas that include aquatic environments such as ponds, lakes, rivers, estuaries, and in fact coastal zones. And in addition to that, until 1970, it was accepted practice to dispose of waste, including excess or obsolete or unserviceable munitions, in deep water areas. Combat operations, as well as accidents, have resulted in munitions being present in the aquatic environment. And underwater munitions are a source of contaminants through either slow leakage or breaches in the munition itself or low-order detonations. And inland freshwater bodies as well as coastal areas can also receive munitions from effluents and runoff from ranges that are on land itself. In the next slide, you see a cross-section of a munition and materials represented within that munition. Because of corrosion, when you have a munition in the aquatic environment, that can lead to breaching that occurs very slowly. Unexploded or discarded munitions in the aquatic environment are either completely or very close to completely filled with the explosive fillers. And if they're exposed to the water, these energetic compounds, such as those that are shown diagrammatically on this slide, such as TNT, RDX, picric acid, nitroglycerin, will slowly dissolve and enter into the surrounding environment. The next slide just gives just one example, highlighting negative public perceptions about these sites. These underwater munition sites around the world have generated a lot of interest within the research community itself, and certainly concern within the communities, public communities around these sites. And while discarded bombs filled with chemical warfare agents such as mustard gas are a focus of much of the environmental and human health concern at these sites, munitions filled with conventional explosives have been considered by some to be a source of significant environmental risk. Considering that underwater munitions are a source of contaminants, site managers may choose to remove them, which is costly, poses human safety risk, and the act of removing the munition itself can also result in release of contaminants into the environment. So it's very important that risk management decisions at these sites be based on sound scientific evidence regarding environmental risk, which is, in essence, the motivation for the investments that our organizations have made over the last 25 years on the fate and effects of these constituents in aquatic environments. So with that, I'll transition to Dr. Lutufo, who will get into the specifics of the work we've done related to this project.
Thank you, Todd. So our primary objective was to look into the risk. In general terms, it could be posed by the release of munitions constituents that we hereby will abbreviate as MC into aquatic environments, having underwater munitions as a point source of contamination. So when we talk about risk, it has two components. Risk is an intersection of, on one side, potential for exposure, the existence of exposure and a quantification of such, and also the potential of hazard or potential for toxicity. And the intersection of those would be an area where risk could potentially occur. So exposure can be characterized by sampling water, sediment, and biota at a site of concern, and also by modeling using known or hypothetical parameters. So examples are shown here on the left. So risk is also more accurately predicted when we know about the distribution of concentrations in the environment, as opposed to, for example, using a more conservative approach, which would be to use the highest concentration detected at a site. And likewise, risk is more accurately predicted, more realistically predicted, if we use toxicity data for a range of species as opposed to using data for the most sensitive species, the most sensitive endpoint derived. So that's what we're going to be showing today, to focus on both of those. And this presentation is based on a very comprehensive and broad overview from our SERDP project, and the title is listed here at the bottom of the slide. So starting by talking a little bit in general terms about the fate of munitions constituents, or MC, in aquatic environments. Using environmental fate parameters collected from the scientific literature, we conducted simulations of the fate of TNT and RDX in a hypothetical water body using a fugacity-based model called quantitative water-air sediment interaction mathematical model. So this model simulation, it overall indicates that most of the mass of TNT and RDX entering a hypothetical aquatic environment would over time become mostly associated with the water column as opposed to the sediment. And because TNT and RDX undergo transformation in water and sediment and also in biota very fast and to a great degree, the residence time of those chemicals in the aquatic environment was estimated to be in the order of days or weeks. So that contrasts, for example, with chemicals with a long residence time and persistence, such as PCBs. So you would need a continuous input of RDX and TNT into the environment otherwise these chemicals and their derivatives would rapidly disappear. So now I'm going to talk a little bit about data derived from another SERDP project. The lead was Dr. Craig Tobias, and they empirically examined the fate of TNT and RDX in aquatic environments using a stable isotope of nitrogen as a tracer and a fairly large-sized mesocosm that simulated intertidal marsh as their model system. So during a period of 2 weeks, there was a continuous flow of water spiked with TNT or RDX entering the mesocosm, and on the other end, there was an outlet for the water. So there was a continuous input of chemicals. And they detected a wide variety of derivatives, including mineralization products. So they really showed that the parent compounds had a very short life in those systems with a lot of transformation going on, sorption to sediment, sorption and transformation into the biota, and eventually mineralization, mostly for RDX. So again, it shows that the aquatic environment does not retain much of this chemical, especially the parent compounds, over time. And so exposure to be prolonged would require continuous input. So like I mentioned, one way to look at exposure when you're conducting or evaluating risk is to take samples at a site and actually measure what's there. But there are also ways to use modeling to determine the distribution of concentrations in the environment. Specifically for underwater munitions, under ESTCP funding, the Navy conducted and produced a model called the Shell model that predicts the release of munitions constituents from breached underwater munitions. So using the Shell model, the release of munitions constituents out of a breached munition, called F in the top equation here, can be estimated using a series of variables, such as the radius of the breach, the cavity radius formed during release, and the prevailing current in the area, and a number of fate parameters related to the MC of concern, such as the solution rate. So when the parameters are known, F can be estimated deterministically using empirical values and information. But when the parameters are unknown, F can be determined probabilistic using hypothetical values. So we're going to look at examples of both. So deterministic evaluation was conducted during a flume study that Gunther will describe in detail in the next presentation, where we had a surrogate munition that was loaded with composition B, which is a combination of TNT and RDX. Into the surrogate munition, there was a cavity and a pinhole that allowed TNT and RDX to slowly enter the flume water during the experiment duration. It was about 10 days. So the measured concentration in the flume water shown in this table here, as in the column labeled empirical, is actually what we measured, was fairly similar to what was predicted by the shell model. So it gave us confidence on using this model for predicting release and exposure in aquatic environments. So the approach for a probabilistic scenario, this was the deterministic scenario, is shown in green here, and was applied to the former Viegas Navy training range. It started by using site-specific information that happened to be available for this site on the number, type of integrity of underwater munitions and current velocities, as well as some hypothetical values for breach size and internal cavity. So it works with distributions. So this graph showed distribution functions used to describe model parameters. And we have a velocity that was centered around 5 centimeters per second, breach holes, and the cavity radius, and so forth. Number of breach shells that was there, for which there was information in the area. And also, it assumed that most of the mass of the munitions constituents was still inside the shells. Todd showed a picture earlier showing a cross-section, and that has been shown to be true for most sites. So using the data, empirical data for the site and hypothetical data for some parameters, quantitative information was derived, such as in terms of a probability distribution and the cumulative distribution. And that's in terms of the mass released from the breach munitions into the water over time. So if one takes information about the volume of water at the site, and in the case of Viegas, it was assumed that there was one full flush of tidal flushing exchange in the water within the bay per day, the model was able to estimate the concentration, the steady state concentration of TNT, that was the MC used for this modeling, in the bay. And it was in the parts per trillion range. And later on, when we conducted sampling at that very site, and Gunther will provide a lot of details on that, it pretty much, that empirical data validated the predictions from the shell model. So in summary, we know that munitions constituents entering the aquatic environment are associated with generally as a low mass, because only the leakage from the munitions is very slow, and then they're subjected to a series of transformations of processes that will lead the parent compound to disappear, and also the derivatives to also disappear or become chemicals that are not considered toxic to the aquatic biota. So that's an overview of the fate of munitions constituents. So now let's talk a little bit about the other side of risk assessment, that is the evaluation or knowledge about hazard. So here I'm showing on a log scale different concentrations for different commonly present in the environment, munitions constituents commonly present in aquatic environment. So for example, TNT, there was a wide range of toxicity because species varied in their sensitivity to that chemical, while others, such as RDX, typically did not produce any effects, even close to solubility. And we had on one extreme MC that were fairly non-toxic, such as nitroguanidine, and on the other extreme, MC that were fairly toxic at fairly low concentrations, such as tetral. But overall, most of the toxicity data fell in the parts per million range, and most of the data fell above 1 part per million. So we derived toxicity data for amphibians, fish, aquatic invertebrates, and autotrophs, and all that has been compiled into a database that will accompany our final report when that is made available. So what typically happens is, one can research a chemical, end up with a tremendous amount of information about that chemical, the toxicity of that chemical. So what regulators like to use are single values that they can use for comparison of like environmental concentrations, right, to assess the potential for risk. So a commonly used value is the acute and chronic water quality criteria. So following procedures from the EPA, one can take the data in the literature and derive these values. So I'm showing here an example for TNT, for freshwater, marine, and also combined water quality criteria, both acute, that's for short exposures, or chronic, that's for long-term exposures. And those values are in the parts per billion range, and they can be used for comparison to actual data from a site for making a screening level determination of risk. Another approach is to derive species sensitivity distributions, where you integrate the data derived for a large number of species and a large number of taxa. So as an example here, each point is the data for a different species, and they're integrated into this curve, and it shows the proportion of species affected at a gradient of concentration. So the lower the concentration, the more species are protected. Right? The higher the concentration, only the most tolerant species are protected. So one can model these and derive benchmark values for toxicity, comparisons to exposure values, and a value that's commonly used is called the HC5, which is the hazard concentration that for 5% of the most sensitive species. So it's derived from this curve. Likewise, for sediment, there's some sediment quality benchmarks out there. Most of you must be familiar with what sediment quality criteria or sediment quality guidelines. And unfortunately, those are not available for munitions constituents. But we have preliminary sediment quality benchmarks that were derived by PASCO in 2010 for use at the Jackson Park site, and he used aqueous toxicity data to derive sediment toxicity data using equilibrium partitioning. So here you have to use this parameter called KOC, that's the partition between water and organic carbon, and predict the toxic concentration in sediment. So here are some examples of those values using the low-end KOC and high-end KOC, because there's some uncertainty associated with those values. So those can be also valuable, but this, as acknowledged by the authors, these are fairly conservative because they compare very conservatively to actual toxicity data that has been derived for actual exposures to spiked sediments, which unfortunately are few in number and don't allow themselves for calculations of sediment toxicity. That's why we have to borrow aqueous toxicity data for this. So now let's move towards talking about actual data from actual sites. So for this effort, we looked at over 30 publications, and we looked at a wide number of sites throughout the world and extracted the data that they reported for concentrations of MC in water, sediment, and tissue. So because of the nature of this webinar, I don't have time to get into detail about some of these sites, but some are, most of them have underwater munitions as a result of the dumping of those munitions after major wars, back then when that was not regulated. For some of the sites, there's a very high number and density of those munitions underwater. And another site, for example, the Viegas site, is different. It was underwater munitions are present as a result of training. So talking a little bit about the data that we obtained for many of the sites, sampling of water resulted in below detection for all the samples, and those sites are listed here on the top table, while for some sites, the water samples resulted in detection, and they were overall fairly low in the parts per billion range for some sites in the parts per trillion range. Of course, the studies were different from each other. They had different objectives, different approaches for sampling, different analytical procedures, so all that has to be taken into account when we're making broad conclusions. But overall, one can see that there wasn't much in terms of concentration. They were relatively low in the water column. So here I'm showing some of this data for two sites, for the Viegas bombing site and for Halifax Harbor. So the top graphs show the frequency of detection, so the number of samples with detection, and the bottom graphs show the actual concentrations on a log scale. So you can see there's, for Viegas, for example, there was detection frequency was fairly low, and Gunther will talk more about that site. And the concentrations were also fairly low in parts per billion or lower. And for comparison, I'm showing earlier data for that site derived by Dr. Porter. By the way, all these references will be presented at the end of this slide, so you can look at the actual papers if you wish to. For both Viegas and Halifax, samples were targeted. They were taken close to munitions that were believed to be leaking. And you can see for Halifax, detection frequency was higher, but concentrations were lower. So, as I mentioned, regulators like to use, risk assessors like to use values such as water quality criteria. So here I have the highest concentration for various sites, presented here in different colors for different MC on the x-axis. And for comparison, I have the chronic water quality criteria. So when the highest concentration at a site is below chronic water quality criteria, there's an indication of negligible or very low probability of risk. And when it falls above, there's some potential for risk, and one should look further into it. So they all fell below here for this MC from various sites, an exception being TNT for Viegas from the Porter study, where he sampled fairly close to a breech munition, where that number was substantially higher than the water quality criteria. So sometimes we have to acknowledge the presence of some hot spots that could occur close to the source, to the leaking. But overall, those sites did not have concentrations in the water column indicative of risk. And we look at sediment, there's a similar situation emerged where detection was for most sites infrequent and only for some MC, and the concentrations were also overall low with some exceptions. Again, the sites may have spots where you close to the source where you can have concentrations that would be high, like high parts per million range, but for the most part they were in the parts per billion range. So similar graphs here showing two examples. One is Jackson Park, it's a shallow area in Puget Sound where you have the presence of small munitions, but a site that also received a lot of contamination from runoff, and you can see low frequency of detection. And also for another site in Hawaii, it was this site for discarded munitions. You can see also a low frequency, and you can see here a broad variability and broad ranges in concentrations for the MC that were actually reported with detectable concentrations. So this is some examples of the data that for which we provide an overview in the report. So, so for sediment, one can do a similar exercise as for water. For water, you have water quality criteria. You have those HC values from the species sensitivity distributions. For sediment, the best we have for the screening evaluations are those values from PASCO that are substantially conservative, but still, and for that reason, when you determine the hazard quotient, which is you can take a concentration in the environment, if you want to be conservative, you use your maximum concentration. You divide that by that benchmark value, and if you are below 1, that's indicative of low potential or negligible potential for risk. If you're above 1, there's some potential for risk, but then you should go into a more detailed evaluation using all the data that's available. So you can see here, looking at the right columns at this table for different sites and different MC, the hazard quotient for sediment was fairly low, rarely being over 1, with one glaring glowing exception here, again, at Viegas, using Dr. Porter's data, where he sampled fairly close, just a few centimeters away from a leaking munition, and he found, according to his report, a hot spot for TNT where risk would definitely be higher. But overall, you can look here at the number of samples. Some of the sites were fairly well characterized. Their hazard quotients were fairly low. So in conclusions, first of all, MC are typically toxic to aquatic organisms at relatively low concentrations. At relatively high concentrations in the parts per million range. But because for some chemicals we have substantial differences in sensitivity among species, some are impacted at the parts per billion range. But overall, most of the toxicity data falls in the parts per million. So that would be milligrams per liter in the water. For sediment would be milligrams per kilogram. So the bulk of the data we compiled pretty much showed that underwater munition sites are contaminated with MC in the parts per billion range. So when we compare that with the toxicity, only a few species should suffer harmful effects of those concentrations. With exposure, higher exposure occurring in close proximity to breach munitions. So despite the challenges associated with characterizing exposures at underwater munition sites, that's going to be the topic of the next presentation, the available data indicates that environmental risks associated with underwater munitions as a source of contamination to the aquatic environment should be low or negligible for most sites when you're considering the whole environment, like if you're looking at the population or community level, as opposed to single organisms that are inhabiting the area fairly close to ammunition. So all this is going to be in great detail in our final report, which we hope will be a valuable resource towards making preliminary assessment of risk using limited site-specific information, which was the objective of our project funded by SERDP. So that concludes the presentation.
Great. Thank you so much, Todd and Guy. And we've received a whole lot of questions for you both. I'm going to get started with Todd here. We'll want to ask you this first question. Given your great introduction about the scope of environmental issues related to underwater military munitions, what elevator speech would you give to an uninformed underwater military munitions site manager? Well,
first, I doubt there's an uninformed site manager, but it would be an interesting elevator speech. I would probably emphasize two things that probably are already apparent to them. There are two major aspects of the work that they're going to be doing, and they take different skills and levels of effort. And one is the communication, important communication challenges and needs that they have in terms of their interaction with both interested and affected stakeholders associated with their site. And a lot of effort and energy on their part is going to be necessary to direct toward that kind of communication to make sure that those interested and affected stakeholders are aware of the work that they're doing and what is going on at the site. The second part, obviously, is the technical issues that concern these sites. And I would want to relay to them that there is a large amount of information available concerning these sites, as has been summarized briefly in this webinar, and that it would be important and very valuable for them and their team to become familiar, intimate with this information from sites all around the world. And as I said at the very beginning, that 25 plus years of science that's been produced on this topic, and to become familiar with that so that they can make informed choices about where they need to make targeted investments in collecting any necessary information in regards to their site in order to make informed judgments and conclusions about what the scope of environmental risks will be at their site.
Thank you so much, Todd. We'll move on to Guy. Here's a question from EPA Region 9. When you discuss the disappearance of MCs in aquatic systems, can you elaborate on whether you're referring to their breakdown or to dilution effect?
Well, I focus mostly on discussing the breakdown. So upon entering the environment, they're subjected to several processes, including phototransformation, but mostly biological transformation and absorption to various matrices that pretty much remove these chemicals into the environment and send them through a cascade of transformation, which sometimes leads to formation of innocuous chemicals such as nutrients, into full or all the way to full mineralization into CO2, for example, or nitrogen. But I didn't focus much on dilution, but that evidently happens as it's an open water environment. Depending on the energy of the environment, dilution will be greater or lesser. If we're talking about an open ocean or deep ocean, you would have a lot of currents. If you're talking about a small lagoon, it could be pretty stagnant with no exchange. So it varies by environment. That's why it's relevant for you for one to construct a conceptual model for the site.
Thank you, Guy. Another question to you. Can you please help us in thinking of a real-world scenario where underwater military munitions could potentially pose high risk to aquatic organisms?
Right. So scenario would be where release would be high, where it would have breach munitions releasing at a higher than typical rate into the water, and the conditions would be such that you would not have much dilution or removal from the area by currents, and you would have a type of sediment and organic and dissolved and or particulate carbon in the water that would not lead to so much transformation. So in those scenarios, release could become the mass release could be more persistent in the environment and lead to higher concentrations that could be harmful to some species. For example, we found that tadpoles are extremely sensitive to TNT. So if you have a small lake that's clean, the water is clean, low carbon in the water, and you have tadpoles present swimming around munitions, maybe there could be some higher risk determined there. But in the bottom of the ocean, that would be less likely.
Thank you. Here's a question from the International Dialogue on Underwater Munitions Foundation, specific to slide 35, which shows some of the data from Hawaii. And the question is as follows. Can you please tell us what you use as benchmarks and whether you investigated low and high density areas?
Well, no, we did not participate in this study. We just obtained the reports, and those were concentrations that were provided in those reports for the sampling effort taken at Ordnance Reef in 2009, a report published in 2014. The reference is it's at the end of the presentation. They provided information about their detection limits and the fact that all the water samples had concentrations below detection. So they did not report any measured concentrations for water at Ordnance Reef. And by reading the report, we learned that they sampled the water using using divers and bottles and they went to sample the water close to munitions that they identified in the environment. So it was a targeted approach, and Gunther will explain more about that approach for sampling, which was applied in Villegas as well for the other SERDP project we'll be talking about today. So this slide, specifically slide 35, it just talks about the results, a summary of the results that's in the report.
Thank you very much. We have a very diverse audience on today's webinar, and this next question is actually from NATO, from the, just a second here, from the NATO's Science for Peace and Security Summer School on Sea Dumped Weapons in Nova Scotia. Can you tell us whether you've researched or investigated the amount of lead in a munitions casing?
No, this project entirely focused on munitions constituents, being explosives that are part of the fillers of those underwater munitions, and their derivatives that they occur in the environment through degradation. So those is what we broadly call munitions constituents. Of course, lead is a military relevant compound, and it could be released into the environment from the presence of underwater munitions and their corrosion, but that was not part of the scope of our project.
And Guy, a related question, can you tell us if similar work has been done for chemical munitions in underwater environments?
There's a lot of work done, and there's a lot of data out there, and it would be nice to have a compilation of the data and an overview similar to ours, but I'm not familiar with any. But the site, one of the sites here, if we have slide 35 up, this sea disposal site Hawaii 05. So I will mention the HUMA project and a report released in 2010, and also a series of publications after that, and more reports on the way. So that's a site where a deep water site in Hawaii where chemical munitions were dumped in high quantities. And despite the challenges, the group of the University of Hawaii and their collaborators went there and took sediment, water, and biota samples. So they have a fair amount of information concerning underwater munition sites focusing on chemical munitions. But I'm not familiar with a project similar to ours where they did a screening level evaluation of risk and summarized the literature on fate and so forth. It would be very valuable to have that.
Thank you, Guy. A question from EPA Region 10. How do biota levels compare to human health cancer and non-cancer risk screening levels for MC?
So, yeah, we focus on the ecological risk here, looking at effects on plants, microalgae, invertebrates, fish. We did not focus on human health, but information is available, and especially for Villegas, there are some reports from ATSDR where they sampled and fish and shellfish, and I think their overall conclusion was to determine that seafood was safe, and they did not attribute any potential human health concerns to the presence of MC in seafood, and I think detection was very, very infrequent in anything that was sampled there, focusing on human risk. So I think available information is seafood is fairly safe for consumption in regard to munitions, and especially if you consider underwater munitions as the source. Of course, you could have other sites where you have other sources of munitions coming into the environment, but in respect to underwater munitions, the information that's out there shows safe consumption of seafood. There are other studies too in Hawaii in that respect. And they send me an email. I'll be glad to send the reports and information that I'm familiar with on this topic.
Right. And as a reminder, the contact information for the speakers is included in the slides, which you can download from the SERDP and ESTCP website. We're running out of time for the Q&A. We are at 23 questions, so we're not going to be able to get to them now. But before we wrap up, 2 quick questions from DTSC and the Navy. Can you please tell us very quickly whether the cumulative risk of metals and explosives was studied? This is a question from DTSC.
I don't think that has been investigated to great extent. I can think of a few studies that looked at interactive mixture toxicity of explosives and metals, or organic compounds and metals. So I don't think there has been much of departure from additivity. But I could also look in more detail on that. That's not something we included in our report. So I would say that overall the studies did not show much potential for synergy, where one chemical would make the other cause a higher effect than by itself.
Thank you.
But there's really not many studies on that.
Okay. Thank you so much. I'd encourage the author of the question from DTSC to reach out to Guy for detailed references. Just very quickly then, question from the Navy. Have you made an effort to disseminate the findings from your study to the regulatory community where you clearly state the low risk to the overall aquatic environment? Have you tried to do that, and how has the regulatory community reacted if you have done so?
We had webinars before, mostly focusing on the data from the next presentation from Gunther, and we spoke with regulators, and some elements from this presentation were included, and they were very receptive. Evidently, the publication of our final report will be the main instrument for dissemination. I hope some of those folks attended this webinar, and I understand anybody can download them from the website and contact us. But we would like to eventually promote a workshop where we can all be in the same room and give a version of this presentation and others in the same topic, and have a discussion about what we know and where do we go from here, and what kind of recommendations can be derived from this effort.
Thank you very much, Guy, and thanks also to you, Todd. And it is now time to move to our next speaker, Mr. Gunther Rosen. Gunther is an aquatic biologist with POCIS in San Diego. His primary research interests are in aquatic and sediment ecotoxicology. For the past 18 years, Gunther has conducted research to improve the Navy's management of contaminated water and sediment. He regularly publishes in peer-reviewed journals and oversees projects related to metals, bioavailability, stormwater impact, in-situ assessment and remediation, as well as underwater munitions. Gunther received a bachelor's degree in aquatic biology from the University of California in Santa Barbara, and a master's degree in aquaculture from Oregon State University. Gunther?
Yes, thank you very much, Rula, and good afternoon, everybody. First, I'm going to just give you a quick overview of the planned talk. I'm going to discuss why we think that we need passive samplers. In this case, I'm going to be talking about an integrated passive sampler called Polar Organic Chemical Integrative Samplers, or POCIS. So these have direct applicability to underwater military munition sites, and I'll tell you why. I'll give you a little background about how they were calibrated for munitions constituents. I'll talk about very briefly a unique positive control field study result, some of our activities associated with refining sampling rates, which are critical to calculating an estimated water concentration from the POCIS samplers, and spend the bulk of the time hopefully discussing field validation, a full-scale field validation at the former Vegas Naval Training Range. So as you already know from the last talk, unexploded ordnance or discarded munitions do wind up in underwater environments, particularly near training ranges, and they pose not only a blast safety risk, but there's increasing regulatory scrutiny associated with whether or not leakage of the explosive fill might have ecological or human health risks. So unlike PCBs or PAHs, which are widespread and persistent, this is a unique scenario where you have small point sources that are all have a variety of parameters that control whether or not explosive fill may slowly leach or leak from the item. So it's a unique situation, and it's hard to quantify and conduct accurate assessments. The POCIS technology, as I mentioned, is a sampler that's been around for about 15 years, and it can continuously samples the water in an integrative manner. So it's not an integrative, or excuse me, it's not an equilibrium sampler like that you often hear about, like SPME or polyethylene, which are used in sediments, but rather integrates over time. And the particular fabrication of the sampler we're using involves Oasis HLB sorbent, which targets weakly hydrophobic organics. Those are chemicals with log KOWs of less than 1 to about 3. So this is a standard off-the-shelf sampler. It was originally developed to look at emerging contaminants, including pharmaceuticals and personal care products. Turns out it works really well for munitions compounds as well, and that was ultimately decided following a Navy NESD project involving calibration. So this is just an example, sort of a conceptual diagram of how one might deploy POCIS at a UWMM site, where you can place samplers in the water column adjacent to items or in more of a grid-style fashion if there's very little information known about the site. I mentioned the Navy's NESD program, which kicked off this effort, which ultimately resulted in the current ESTCP project. And during that early efforts, we had to derive sampling rates, which are molecule-specific, to calibrate for various common MCs. So this list here incorporates what we thought would be the most common munitions constituents that would be found at these legacy sites where conventional munitions were used. So on the figure here, you're looking at uptake of trace levels of TNT and several different degradation products of TNT by the sampler over time. And the study validated the fact that we are seeing linear uptake up to a period of 28 days. And the data also were used to, using this equation, calculate a sampling rate specific to each molecule. You need that sampling rate in order to calculate your estimated time averaged water concentration in the field. So once we had our sampling rates, we were able to go to the field. In this case, quite a unique opportunity because of the uncertainties at actual naval training ranges or DoD training ranges in terms of where the items are and which ones are leaking at any given time. We had the opportunity to go to a clean site at EPA's Gulf Ecology Division in Gulf Breeze, Florida, and with various permits, place a small quantity, specifically 15 grams, of composition B, which, as Guy noted, is a mix, a fill material. It's a blend of RDX and TNT with wax, and place this at a designated location right here. The yellow dot there is where we place a the 15 grams in a source canister, basically inside of a POCIS canister, about a foot above the bottom. And then we proceeded to add a series of concentric circles incorporating passive samplers, oysters, and grab water samples for a 2-week period. So real succinctly, after 2 weeks, about 1.5 grams to 2 grams of the 15 grams had been lost to the environment. So Comp B does dissolve very slowly, so we were concerned we might not get a detect. But we did for both TNT and RDX, and we observed what you would expect, which was the highest concentrations immediately adjacent. We're talking less than a foot away from the source canister, but the detectable concentrations only up to about 2 meters. At our 5-meter sampling point, we were at non-detect levels. Concentrations that were detected were in the range of 9 to 103 parts per trillion, or nanograms per liter. And if you recall Guy's talk, those are pretty small concentrations with respect to toxicity. Incidentally, our grab water samples, which were 1-liter grabs taken at several points during the 2-week period, and our tissue samples were all below their associated detection limits. This isn't really surprising considering the relatively low propensity for most of these chemicals to bioaccumulate, and also the detection limits for standard grab sampling. Another effort that took a considerable amount of time, multiple experiments, was to look at optimizing the POCIS sampling rate. So the sampling rate is affected by various factors like biofouling, temperature, and in particular flow velocity across the membrane. So we looked at this at the ERDC lab in Vicksburg, Mississippi, using a large flume, a 30,000-gallon two-sided flume. We spiked the flume with a cocktail of various munitions constituents at a part per billion. And with precise control over flow and monitoring that flow rate, we were able to quantify uptake rates across different scenarios, ranging from 7 to 30 centimeters per second. We were also able to validate the shell model that Guy brought up as well. In the picture on the right, we have a simulated breached munition with Comp B fragments, and we had a few different scenarios where the fragments were either fully exposed, similar to a low-order detonation, or just through a pinhole kind of a breach as well. So I can't go into all the details here on those studies, but what we found was that TNT and DNTs, dinitrotoluenes, do have flow dependence, and sampling rates differ by a factor of about 2. RDX was less affected by flow rate, with ranges up to about a factor of 1.5. But from these data, we are able to calculate, derive equations to make a more accurate estimate of munitions compounds if you measure the flow velocities at your site. So when you go out to your site, you want to deploy POCIS, but you also want to deploy a current profiler or some sort of current meter. So the remainder of the talk here is going to focus on our field effort at Vieques, Puerto Rico. As you know by now, this is a former naval training range, and we focused on the east side of the island, known as the live impact area. This area involved air-to-ground bombing and gunfire from 1941 until its closure in 2003. And our site was Bahia Salina del Sur, which is a small embayment on the southeast end of the island. And I need to point out that we were authorized to do this work by Dan Waddell and his NAFAC remedial project managers. Their involvement was critical to the success of this effort, and they provided a tremendous amount of in-kind and very talented support to get the job done. So we targeted two different types of approaches to demonstrate the value added of using POCIS in a monitoring program. And one was a grid kind of a design where you presumably know very little about where items are and what their condition is, and then also a much more targeted, focused approach. So in order to identify items, we had to first do a reconnaissance survey. So the CH2M Hill Munitions Response Dive Team went out a few days before our deployment and identified 25 items, from which we selected 15 to look at. And these involved a variety of types of munitions, ranging from MK series bombs to 5-inch projectiles, and also various condition. Some of them appeared to maybe be low-order detonations, while others looked like they were fully intact. Some were buried. And you can see here on the top left the strategy for deploying the POCIS. We can't suspend these from a dock or anything in this case, so we have this weighted block system. On the top right, we have in areas where we were using the grid style and we weren't necessarily adjacent to a munition, we were able to use sand screws to hold the POCIS intact. This shows the overall test design. We had a team of 5 divers every day on the boat. It was a considerable effort. Each station had to be swept with a magnetometer to make sure it was safe. Then we would proceed to deploy, whether on the block or with the sand screws. This was followed by scientific divers collecting grab water samples adjacent to the items. So grab water was collected at 15 target stations, not at the grid stations, both at the beginning of the study and then on the recovery end as well. And these items at the target stations, we were able to position the samplers within 6 to 12 inches from the target item, and we targeted a breach if we saw one. Because of the tendency for TNT and other nitroaromatics to degrade quickly, we had to use on-site water extraction, and then those samples were shipped to the analytical laboratory. And Jason Belden, one of the project team members, used EPA 8095 modified for GCMS with negative chemical ionization, and you can read about his work he's done in his paper attached. So ultimately, this was the distribution of our total of 30 POCIS samplers deployed at Bahia Salina del Sur. In the green are our grid stations, and the red are our target stations. And you can see sort of a cluster of them along the coastal areas, and then 2 at the south end, outside the bay, south of what's known as Roca Alcatraz. And just a real quick summary of some of the highlights of those data. For one, we were very pleased that both deployment approaches worked. That was a big relief for us, and 100% of our samplers were recovered intact. As far as chemistry goes, TNT and TNT degradation products were only quantified in 1 of the 15 targets samples at concentrations above the reporting limit. This was adjacent to a 1,000-pound general-purpose bomb. Divers had characterized several visible breaches at this item, and we detected, both with grab samples and POCIS, low part per billion or microgram per liter concentrations. So we're talking about 5 parts per billion, which is about 2 orders of magnitude below where you might see any potential to exceed that HC5 that Guy referred to. And what I found interesting here was that the grab samples and the POCIS, if you take the average of the two grab samples, they're pretty much the same value. And this suggests to me that this was probably a continuous source during the 2-week period. And the fact that we measured primarily TNT, we also did measure degradation products, but the fact that about 90% of it was TNT suggests that it was a continuous, fresh release. So very few TNT hits. For RDX, we saw more detection, in fact, with POCIS at a frequency of 79%, but these were at extremely low concentrations, usually single-digit part per trillion concentrations. Grab samples we detected much less frequently. And this just shows the distribution here for RDX. Concentrations it didn't really seem to matter whether or not we were at a grid station or at an item. But what we did notice was that concentrations of RDX tended to be detected more along the coastal areas as opposed to the inside of the bay. But above the reporting limit, only 4 out of the 29 stations resulted in detection. Just so look from those RDX data, if you look back at the species sensitivity distribution that Guy described, this is for RDX, NOFX concentrations. RHC5 is 6 orders of magnitude higher than the concentrations we were measuring at the Bahia. And again, for TNT, we were 2 orders of magnitude lower than the HC5. So we went back to a select number of stations, where we did see RDX hits to look at how our POCIS data compared with any detection in sediment or sediment pore water. We used push point samplers very close to within a half a meter of several items, as well as at a farther distance. And in the spirit of time, I'm just going to skip to that result. These are not hits. This is just a representation of the method detection limits and the quantitation limits. All the samples were below method detection limits, so for both pore water and sediment. So in this case, POCIS was the most informative and conservative measure. So we are right now finalizing our project report for ESTCP. It should be submitted within the next couple of weeks. That'll be attached. Attached to that will be an end-user guide that we think will be very helpful to the community. The samplers are already commercially available, so people can use POCIS at their sites at any time. Again, the interactive nature of the VACUS effort was an extremely great opportunity to get end users, site managers aware of the technology. They seemed to understand what it was for and saw the benefits. Similarly, our Gulf Reef study on the premises of an EPA laboratory, Dr. Mace Barron expressed a lot of interest in the technology. So that's another type of hopefully visibility for the regulatory acceptance aspect. We've provided a number of webinars to the DoD community, and there are several publications in press and in preparation. So just to conclude, I think that POCIS was successfully validated for assessing MC exposure at underwater military munitions sites. Advantages are its high sensitivity and its potential to integrate over time, especially where you have potentially variables that, such as leakage or tides, that might affect the value of a grab sample by potentially potentially sampling at the wrong time or at the wrong place. The positive control and VIECAS site results validated laboratory studies that we had conducted over several years prior, suggesting an overall message of highly localized or negligible exposure and low potential for ecological risk. And the benefits of the DoD, we feel, is more accurate assessments resulting in or contributing to appropriate management decisions, whether to leave in place or potentially remove munitions. Again, the cost analysis and the user guide are in process and will be up on the ESTCP website soon. And I think I'll leave it at that. Thank you very much.
Thank you so much, Gunther. We have received a number of questions. Are you suggesting that polar organic chemical integrative samplers would replace grab sampling?
I think that they are another tool in the toolkit, and it would depend on the objectives of the site and the regulatory requirements. So it would be very site-specific. There's clear advantages to using them, and just like passive samplers for contaminated sediments, there's a large thrust towards getting them more commonly integrated into regulatory programs. I don't think that they would necessarily always replace a grab sample.
All right. And then how does the cost of these POCIS samplers compare to that of grab samples? And how commercially available are these samplers? Can you just go out and buy them?
Yes, there's a website, environmental ESTinc.com sells the samplers. So they've been around for a number of years. People are using them for all kinds of monitoring programs. So they are easy to get. The cost analysis, I'm working up the cost analysis right now, and it depends on how your grab sample is going to be collected. If you're going to go out to a site and collect a single grab and compare that cost with a deployment and recovery operation of a POCIS, you're going to be spending more on the POCIS. But if you want something comparable, you would really need multiple grabs. And so the costs actually begin to quickly become in favor of the POCIS, just depending on the number of samples you need to make a composite that's representative of what's actually occurring at the site.
Thank you. How will you make the cost analysis results available?
That will be part of our ESTCP final report, and that will be available as soon as we can get it finalized in the next couple of months. And we're outlining several different scenarios because of the complexities of trying to compare grab samples or auto composite types of samplers to POCIS, because they tell you two different things.
Thank you. A question from the Navy. Given the analytical methods that you use, how confident are you that the detections were representative of the actual MCs and not interference?
I think we're very confident because Jason, Jason Belden, who's the chemist on the project, provided quite a rigorous QA/QC of the samples, collected field blanks, and spent a lot of time trying to make sure that his peaks were real. So we're pretty confident.
Thank you. A question from EPA Region 10. Could POCIS be used to assess long-term dissolution over a period of 50 years or more of MC for in-place munitions?
Yes, but you'd have to switch out the POCIS. You wouldn't want to just put one there and leave it for 50 years. We find that after a month, you start running into issues with biofouling, which will impact the sampling rate. So you could go out and replace the POCIS and then have a time sort of series over a long period of time, just continue to replace them.
Thank you, Gunther. And one very quick question before we pull Todd and Guy back in. This is a question from EPA Region 9, and it relates to the reporting limits that you're referring to. Can you elaborate on whether these are reporting limits specific to a method or a group of contaminants or to a regulatory value?
Yeah, I may let Guy weigh in on that when you bring him back on, but the reporting limit is, in this case, is 3 times the method detection limit, but that's all I can really offer.
All right, well, this is a good question to pull you back in, Guy. Can you please weigh in?
Can you please repeat the question? I heard the answer that Gunther said, but yeah, please repeat.
Yeah, this is a question from EPA Region 9, and it relates to the reporting limits that both of you referred to. Are these reporting limits specific to an analytical methodology that is EPA approved or a research-based method, or are they related to, are they compound-specific reporting limits? Can you just give additional detail on the reporting limits that you both referred to?
Yeah, I referred to below detection limits in a table, and those were reporting limits or quantitative limits as found in the reports. Most of those studies were pretty rigorous and followed EPA guidance for deriving them, not all. So there's some variability among studies. But the major studies where we derived most of the data, they were rigorously derived using, there was some quality control information in the reports.
Thank you. Now a question for both of you, and Todd, I'd like you to start answering. How do the newer generation insensitive munitions compare in toxicity and presence at underwater military munitions sites?
Well, I think probably Guy's in a better position to answer that question just because of the work that's been going on by Guy and others in this area. Guy, do you have thoughts about that?
Yeah, I'm having trouble hearing Rula. You said something about generation. I'm sorry. Can you please repeat? I'll put my volume as high as it goes.
Okay. The newer generation insensitive munitions.
Oh, yes.
Yeah.
Yes, now new generation insensitive munitions. I thought you were talking about age groups. The new generation sensitive munitions, overall, they tend to have lower toxicity than conventional munitions. And it's quite variable. One of the chemicals called DNN, it has a somewhat similar fate in the environment as TNT. Others are components of insensitive munitions are also used in conventional munitions such as nitroguanidine that has fairly low toxicity, but it also can become photoactivated in the environment and cause higher toxicity. So, but in terms of underwater munitions, it's really not an issue because they're not there as munitions. You know, they're present in the environment from training and testing mostly and production effluents, but they're not really packaged into munitions that were dumped or made their way into the underwater environment. So now, you know, there's a lot more regulations and care into those. So there should not be a similar legacy scenario for future generations of people dealing with them, like we're dealing now with what was done after the First and Second World War. You know, tremendous numbers of munitions dumped into the waters throughout the world. That's not going to happen with sensitive munitions that are being developed now.
Thank you so much. And Todd, you spoke about the attention from the media and from the public on underwater military munitions sites. And it would appear that the public has a negative perception about these sites. So what suggestions do you have for DoD in general and other stakeholders to address such perception issues?
Right. And by perceptions, I would never want to imply that, there's no basis for interest or concern. Clearly, this is, concerns are legitimate. We have materials in the environment and questions about what the consequences or effects or risks, either in the short or long term, associated with those materials are legitimate questions to ask. I think, as I started out by saying, organizations like ERDC, the Army, and the Navy have made sizable investments over, a quarter of a century, 25 years, looking at the environmental implications of these, unique constituents. I mean, there are a limited number of organizations that are concerned with and have been investing so much attention in the science associated with this unique set of chemicals. So the Navy and the Army, have taken this seriously, as reflected by the amount of investment that's been made, and certainly SERDP and ESTCP have been leading in that area. But I think we're at a point now with all of the data that have been generated where more interest and attention needs to be given in communicating what has been learned. Scientists like myself and Guy and Gunther, we publish work in the scientific literature, but of course that's only going to be read by a narrow slice of the broad community that is interested and concerned about these sites. So broadening the communication, which is two-way, broadening the conversation about these sites and introducing what has been learned using these data, this science, this evidence, I think is a necessary next step to focus on these currents and hopefully make progress toward finding resolution or finding, sustainable solutions for these sites that exist around the world.
Thank you, Todd. Ian, Gunther, would you like to add to this discussion before we wrap up? Feel free to jump in.
I think Todd did a really nice job at answering that question.
Yeah, communication would be definitely key because it can be really overwhelming. If you know that not where the place where you get your seafood from, your fish from, or not too far from there, there is this tremendous amount of munitions encased in metal, and corroding. Definitely, there's room for concern there. But so one needs to kind of zoom out from that scenario and look at the broad picture. What do we know, and really understand the concept of risk. It's exposure divided by hazard. So understanding that and looking at graphs, for example, the one that Gunther showed, okay, exposure is at this far end, hazard is at this other far end. Oh, okay, so understanding the basics would definitely help understand the problem.
Great. Thank you so much. All three of you have done an outstanding job, and for that we are grateful. Before we wrap up, I'd like to remind our audience that the next webinar is in 2 weeks on April 6th, and it will focus on 1,4-dioxane impacts and innovative cleanup technologies at DoD contaminated sites. The webinar will feature 3 speakers: Dr. Hunter Anderson from the U.S. Air Force Civil Engineer Center, Dr. Shelley Mahendra from the University of California at Los Angeles, and Dr. Robert Hinchey from Integrated Science and Technology. So registration is already open for this webinar. Feel free to visit the SERDP and ESTCP webinar webpage to register for this and other future webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it if you can please take a brief moment of your time to complete the survey that will pop up on your screen at this time. This concludes today's webcast. Thank you for your attention.